Good morning, and welcome back to this channel, the place where real science meets real life for seniors who refuse to simply accept aging without a fight. What if I told you that sitting right now in your kitchen, possibly less than 10 feet away from where you're watching this, there is something so simple, so affordable, and so backed by real science that it could genuinely influence the biology of your aging body? Not a pill, not a prescription, not some expensive supplement costing $300 a month, something you can add to your morning coffee starting today. And before you roll your eyes and think you've heard this kind of claim before, stay with me, because what I'm about to share is not a trend or a gimmick. This is research that's been quietly building for nearly 2 decades, with a Nobel Prize attached to part of the underlying mechanism, and most people simply haven't heard about it yet. If you're over 50, this is genuinely worth your full attention. I'm Dr. Marie Huberman, and on this channel, we talk about real science in real language, because you deserve to actually understand what's happening inside your own body. Today, we're counting down through the cellular biology behind one specific compound found naturally in your food that researchers studying stem cells metabolism and cellular aging have been investigating with real, published, peer-reviewed seriousness. Stay with me, because this gets genuinely interesting fast. Drop a comment below telling me if you've ever heard the word autophagy before today, and hit like and subscribe if you want more of these deep dives into the actual cellular science behind aging, because that's the entire point of this channel. Let's start with the big picture. By the time most people reach their 60s, something has quietly shifted at the cellular level. Your body contains trillions of cells, and among them are what scientists call adult stem cells, your body's own internal repair crew. Think of them like maintenance workers in a massive building. When tissue is damaged or organs begin to wear, these stem cells are supposed to activate, replicate, and fix the problem. But here's what most people don't realize. As we age, those stem cells become progressively less responsive. Research in the field of stem cell biology has shown that aging stem cells often don't disappear outright, but the surrounding cellular environment becomes less hospitable to their activation, meaning the biological signals that once triggered repair become weaker over time, while background inflammation tends to rise. This is part of why a person in their 60s typically heals more slowly from an injury than someone in their 20s, and why chronic conditions tend to accumulate gradually rather than appear overnight. It's worth understanding a little more about why this happens, because it isn't simply that stem cells wear out the way an old battery might. Researchers studying what's sometimes called the stem cell niche, the specific local environment surrounding these cells, have found that the chemical and physical signals in that environment shift considerably with age. Some of this involves changes in the extracellular matrix, the structural scaffolding that surrounds cells throughout your tissues, and some of it involves shifts in nearby immune cell behavior, which can either support or actively interfere with stem cell activity depending on the local inflammatory state. This is part of why simply isolating stem cells and injecting them elsewhere, an approach some commercial clinics have tried to market, hasn't generally produced the dramatic results some have hoped for. The cells themselves are often perfectly capable, but the environment they're being placed back into may still be sending the wrong signals. This distinction matters a great deal for understanding why a strategy that works at the level of the broader cellular environment, rather than trying to replace or transplant cells directly, has generated so much genuine scientific interest in recent years. But scientists have also discovered that certain naturally occurring compounds appear capable of supporting and reactivating parts of this dormant repair signaling, which is genuinely one of the more hopeful threads running through modern aging research. Before we get to the compound itself, there's one more piece of the puzzle worth understanding, because it makes everything else click into place. It's called autophagy, a word with Greek roots that literally translates to self-eating. I know that sounds unsettling, but it's actually one of the most remarkable and beneficial processes your cells are capable of. Autophagy is essentially your body's internal housekeeping system. When activated, your cells begin breaking down and recycling damaged proteins and malfunctioning internal structures called organelles, clearing out cellular debris that would otherwise accumulate and contribute to dysfunction over time. A researcher named Yoshinori Osumi won the Nobel Prize in Physiology or Medicine in 2016, specifically for his work uncovering the genetic mechanisms behind autophagy, using yeast as his initial research model before the findings were shown to apply broadly across more complex organisms, including humans. This isn't fringe science. It's Nobel-recognized cellular biology, and the research building on it over the past decade has only grown more substantial. To put it in slightly more practical terms, picture your cells as a busy household that's constantly generating clutter, worn-out furniture, expired food, broken appliances. A well-functioning autophagy system is like having a thorough cleaning and recycling crew come through regularly, sorting out what's salvageable, breaking down what isn't, and keeping the whole space functional. When autophagy activity declines, that clutter starts to pile up instead, and the cell has to operate in increasingly cramped, inefficient conditions. Multiple studies have linked declining autophagy activity to accelerated cellular aging and the buildup of metabolic dysfunction, including the kind of stubborn fat retention that seems resistant to diet and exercise alone. Researchers have coined the term inflammaging to describe how chronic, low-grade inflammation appears to function as one of the central biological drivers of the aging process itself, and the relationship between autophagy, stem cell signaling, and inflammation is now understood to be deeply interconnected. When one of these systems falters, the others tend to follow, and a meaningful number of people are functioning every day with all three operating well below their potential capacity, often without realizing it. This is where your morning coffee comes in. The compound at the center of this conversation is called spermidine. Yes, the name does raise eyebrows, since it was first isolated from human semen, which is where it gets its name, but it's present in virtually every living cell across plant and animal life. And importantly, it's found in meaningful concentrations in specific everyday foods. One of the richest and most accessible dietary sources is wheat germ, the nutrient-dense embryo portion of the wheat kernel that gets stripped away during the refining process that produces white flour. Wheat germ is among the most concentrated, naturally occurring dietary sources of spermidine that researchers have identified, with a tablespoon of raw wheat germ generally estimated to contain somewhere in the range of several milligrams of the compound. A growing body of research, including both animal studies and large-scale human observational research, has investigated whether regular dietary spermidine intake is associated with measurable health outcomes, and the findings so far are genuinely compelling, while still leaving some open questions that I want to walk through honestly rather than oversell. One of the most well-known studies in this area comes from a long-running research project in northern Italy known as the Bruneck Study, where researchers tracked dietary habits and health outcomes in a population over a multi-decade follow-up period. Analysis from this cohort, published by researchers in the journal Nature Medicine, found that participants with higher dietary spermidine intake had significantly lower rates of cardiovascular disease and all-cause mortality compared to those with lower intake, and the relationship persisted even after researchers statistically adjusted for general diet quality and other lifestyle factors. Separate analysis using United States National Health Survey data, covering tens of thousands of participants, similarly found that higher dietary spermidine intake was associated with reduced cardiovascular and all-cause mortality risk, with the protective association showing up most clearly for spermidine sourced from vegetables, whole grains, legumes, nuts, and cheese specifically. That kind of consistency, showing up across genuinely different populations and study designs, is exactly the kind of pattern that gives researchers more confidence that an association is meaningful rather than coincidental, even though, as with any observational research, it can't fully prove cause and effect on its own. So how does it actually work at the mechanistic level? When spermidine enters your cells, it functions as what researchers call an autophagy inducer, essentially nudging open a molecular pathway that's often partially restricted with age. Specifically, it inhibits an enzyme called EP300, which under normal conditions acts as something of a brake on the autophagy process. When EP300 activity is reduced, the cellular machinery responsible for autophagy becomes more active, scanning for damaged components, including dysfunctional mitochondria, the small structures responsible for generating cellular energy and clearing them out for recycling. Animal research, including a well-cited study published in Nature Medicine in 2016 by a research team led by Frank Medeo and colleagues, found that spermidine supplementation extended lifespan in mice and reversed certain age-related changes in heart function, with the protective effect disappearing entirely when researchers genetically blocked the animal's capacity for autophagy. That detail matters enormously because it's strong evidence that autophagy itself, not some unrelated side effect, is actually responsible for the benefit observed, which is exactly the kind of mechanistic confirmation that separates a real biological finding from a coincidental correlation. Human biology is, of course, considerably more complex than a laboratory mouse, but many of the underlying cellular pathways involved are evolutionarily well conserved across species, which is part of why researchers in the aging field have taken substantial interest in spermidine's translational potential. Now, I want to address the cancer connection directly, and I want to do it carefully because this is genuinely one of the more nuanced parts of the research, not a simple, one-sided story. Cancer cells often display a distinctive metabolic pattern, frequently relying heavily on glucose fermentation even when oxygen is available, a phenomenon first described nearly a century ago and now commonly known as the Warburg effect. Because autophagy plays a role in cellular quality control, including the clearance of cells that have started accumulating dangerous mutations, researchers have investigated whether boosting autophagy through spermidine might support the body's natural surveillance against early cancerous changes. Some research, including animal studies on liver and oral cancers, has found that spermidine-induced autophagy appeared protective, reducing tumor formation and supporting DNA repair processes. But the honest, complete picture is more complicated than a simple cancer-starving narrative. Polyamines, the broader chemical family spermidine belongs to, are also genuinely necessary for normal cell growth and proliferation, which means some researchers studying this area have explicitly described spermidine's relationship to cancer biology as two-faced, capable of either supporting the body's defenses against early malignant cells in some contexts, or, theoretically, supporting cell growth more generally in others, depending heavily on the specific tissue, timing, and existing health status involved. Reviews on this exact topic, published by researchers actively working in polyamine and cancer biology, have been candid that this remains an active and evolving area of investigation, rather than a settled conclusion, and that's the honest place I want to leave this part of the conversation, rather than promising you a tidier story than the actual science currently supports. Let's talk about the fat metabolism angle now, because I know this is something a lot of people watching have struggled with directly. As we age, mitochondria, the energy-producing structures inside our cells, tend to become less efficient, generating more byproduct molecules called reactive oxygen species, which contribute to oxidative stress and low-grade inflammation throughout the body. This decline in mitochondrial efficiency is considered one of several contributing factors behind the gradual metabolic slowdown many people notice as they get older. A specific subtype of autophagy, called mitophagy, is responsible specifically for identifying and clearing out damaged mitochondria, allowing them to be replaced with newer, more efficient ones. When this process functions well, cellular energy production tends to improve, which is one of the proposed mechanisms behind some of the metabolic benefits observed in spermidine research. It's worth pausing on why this particular mechanism gets researchers genuinely excited, beyond the fat metabolism angle specifically. Mitochondria aren't just energy factories, they're also deeply involved in regulating cellular stress responses, and even, in certain circumstances, in signaling a damaged cell to safely shut itself down before it can cause further problems, a process distinct from but related to the cancer surveillance we discussed earlier. So when mitophagy is functioning well, you're not just getting a metabolic benefit in the sense of feeling more energetic, you're also supporting a quality control system that touches on several other aspects of healthy aging simultaneously. Animal studies have shown that spermidine supplementation can improve markers of mitochondrial function and reduce oxidative stress in aging tissue, And while large-scale human clinical trials specifically measuring body composition outcomes are still relatively limited, the early research direction is genuinely promising enough that several research groups are actively pursuing more rigorous human studies right now. I'd also gently caution against expecting this to function like a dramatic metabolism-boosting intervention in the way some supplement marketing tends to frame compounds like this. The realistic expectation, based on the current evidence, is a modest, gradual support to underlying cellular efficiency over a sustained period of consistent intake, not a quick fix for stubborn weight on its own. So, practically, how would you actually do this? It's simpler than it might sound. Raw or lightly toasted wheat germ, widely available at most grocery stores and health food markets for just a few dollars, has a mild, slightly nutty flavor that blends easily into coffee. A tablespoon stirred directly into your cup, or blended in if you use a frother or blender for your morning brew, is generally close to undetectable in a reasonably strong coffee. Some people prefer adding it to the dry coffee grounds before brewing, while others mix it into a morning smoothie instead. What matters most, based on how this research is actually structured, is consistency rather than a single dose. The observational benefits associated with spermidine intake come from sustained, regular dietary patterns over years, not a one-time addition to your morning routine. It's also worth knowing that wheat germ isn't your only option here. Aged cheeses, mushrooms, green peas, whole grains, legumes, and fermented foods like natto all contain meaningful amounts of spermidine as well, and getting it from a variety of sources across your overall diet is likely a more robust long-term strategy than relying on any single food. Now, here's a quick but important reminder before we move further. If this information has been genuinely useful so far, take a moment right now to comment below with which food source you're most likely to try adding to your routine. Hit that like button and subscribe if you haven't, because that's what helps this kind of carefully sourced content reach more people instead of the louder, less accurate version of this topic that tends to dominate search results. Before we move to where the science currently stands, I want to spend a moment on practical safety, because this is a question I genuinely get asked often. Dietary spermidine from whole foods like wheat germ, legumes, mushrooms, and aged cheese has a long history of human consumption, since these are, after all, just ordinary foods many people already eat in some quantity, which is a meaningfully different safety conversation than a novel, concentrated supplement might raise. That said, a few practical notes are worth knowing. Wheat germ does contain gluten, so if you have celiac disease or a gluten sensitivity, it's not an appropriate source for you. Though the other spermidine-rich foods I mentioned, like mushrooms, green peas, and aged cheese, are perfectly fine alternatives. Wheat germ also has a relatively high fat content for a plant-derived food, which means it can go rancid faster than you might expect if it's not stored properly. Keeping it refrigerated after opening and checking for any off-smell before use is a simple, sensible habit. If you're considering a concentrated spermidine supplement rather than whole food sources, I'd encourage some caution and a conversation with your doctor first, since most of the rigorous safety and dosing research has been built around dietary intake levels rather than the higher concentrations sometimes found in supplement products. And the long-term safety data on those higher doses in humans is considerably thinner. As with most areas of nutrition science, getting a compound from a whole food alongside its full natural context of fiber, other micronutrients, and a moderate food-based dose tends to be the safer and better studied starting point compared to jumping straight to an isolated concentrated form. It's also worth being clear-eyed about who should be more cautious here. If you are currently pregnant or breastfeeding, if you have a history of certain hormone-sensitive conditions, or if you're managing any active cancer diagnosis, this is exactly the kind of dietary change worth specifically running by your doctor first, given the genuinely mixed and still evolving research on polyamines and cell growth that we touched on earlier. For the average, otherwise healthy adult, simply looking to modestly increase their intake of a handful of common foods, the risk profile here is about as low as you'll find in this entire category of health content, but low risk for most people and no need to ever ask your doctor about anything aren't quite the same statement. And I want to be precise about that distinction, rather than blur it for the sake of a cleaner ending. I want to be transparent with you about exactly where the current science stands, because I think you deserve that honesty rather than a more dramatic-sounding summary. Much of the human research on dietary spermidine remains observational in nature, meaning it shows a strong and consistent association rather than definitive, randomized proof of direct cause and effect in every outcome being discussed. The animal research is considerably more mechanistically detailed, and some smaller human trials, including research on spermidine supplementation and markers of cognitive function in older adults, have shown encouraging early results. But larger, longer human trials are still very much needed before we can speak with full certainty about the magnitude of benefit a person might expect. What we can say with real confidence is this: the underlying biological mechanism is well documented and Nobel recognized at its core. The safety profile of dietary spermidine from whole food sources appears excellent based on existing research. The cost of incorporating more spermidine-rich foods into your diet is minimal, and the existing evidence base, while still developing, points in a genuinely encouraging direction. I also want to speak directly to anyone watching this who is currently managing a cancer diagnosis themselves or supporting a loved one through one. Please don't treat anything discussed in this video as a substitute for oncological care. What we've covered today relates to the body's general cellular surveillance and metabolic biology, not cancer treatment. And if you're currently undergoing any form of cancer treatment, it's essential to speak with your oncologist before making any significant dietary changes, since your medical team understands the specifics of your individual situation in ways no general health video possibly can. For everyone else watching, particularly those of you thinking seriously about your energy levels, your metabolic health, and what the next decade of your life might look like, I want you to walk away from this video with a sense of agency rather than alarm. The science of cellular aging is one of the most active and rapidly evolving fields in medicine right now, and what's emerging from it paints a picture of aging that is considerably more responsive to everyday choices than many of us were taught to believe even a decade ago. None of this means aging stops or reverses. It doesn't, and I'd be doing you a disservice if I implied otherwise. What it does mean is that some of the cellular processes underlying how we age appear to be more adjustable at the margins through everyday dietary choices than older models of aging biology assumed. That's a meaningfully different and, I'd argue, more honest message than either extreme you tend to see online, the fatalistic nothing you do matters framing on one side, or the this one trick reverses aging framing on the other. The real picture sits somewhere more modest and more interesting in between. A tablespoon of wheat germs stirred into your morning coffee is a small, low-cost, low-risk place to start engaging with that science directly, today, in your own kitchen. If you decide to try it, give it real time before judging whether it's doing anything. The research we've discussed today is built on sustained intake over months and years, not days. So this is a long-term dietary habit to evaluate the way you'd evaluate any other gradual lifestyle change, not a short-term experiment with an immediate, dramatic before and after. If you found this valuable, and I genuinely hope you did, go ahead and hit that like button right now, since it helps signal that this kind of carefully sourced, real science health content is worth sharing with more people who could use it. And if you haven't subscribed yet, now's a great time, because every week on this channel we dig into research your doctor likely doesn't have time to walk you through during a 15-minute appointment, even though it's exactly the kind of information you deserve to understand about your own body. Before you go, drop one more comment for me. What's one small change you're willing to try this week based on what we covered today? I read every single one, and it genuinely shapes what we dig into next. I'm Dr. Marie Huberman. Take care of yourselves, take care of each other, and I'll see you in the next video. This is your Senior Health and Well-Being channel, reminding you that it is never too late to support your body, honor your health, and live fully at every age. Here's to your best years, not behind you, but unfolding right now.